Alright, guys, I am back. I answered my call from my kid, wondering if it was something, important, urgent. She wanted me to bring her an icy from Fleet Farm. So, there you go. Gotta love kids. Alright, let me pick up where we left off, talking about forming glycosidic bonds between saccharides, between monosaccharides. Alright, so remember what we talked about earlier, right? Looking at the OH group that's on the anomeric carbon, it could be pointed down, in which case we refer to it as being in the alpha position. If it's pointed up, then it's in the beta position. That means we can make what we call alpha linkages and beta linkages. All depends on whether we use an alpha or beta OH, right? As you might expect, if the linkage results from a beta OH, you have a beta linkage. If it results from an alpha OH, you have an alpha linkage. All right, so here's an example of a disaccharide. All right, so you see here, you have these two monosaccharide units, and you have basically the OH of one anomeric carbon. That's from the ring on the left. All right, the anomeric carbon of that ring is linked to another carbon from another saccharide ring by an oxygen. All right. Sorry, my kid's not sending text messages. Okay, so that's a glycosidic linkage. I want you to pay close attention to the way it's drawn, right? If you look over here, the direction of the bonds from these two carbons linking to the O is very intentional. They're pointed down. That's an important notation to make. When they're pointed down like this means that the oxygens from the respective carbons were pointed down. And this is referred to as an alpha parentheses 1,4 linkage. All right? And you'll see this terminology frequently when you're looking at descriptions of saccharide linkages in disaccharides, trisaccharides, what have you. Here's what it means. All right? By definition, the anomeric carbon is number 1 in the ring. Right? Anomeric carbon is always going to be number 1. So when you see the 1, that means the ring on the left, the saccharide on the left, is making a linkage from its anomeric carbon. The alpha tells you that OH at the anomeric carbon was in the alpha position. And then the 4 tells you it was linked to carbon number 4 on the next ring. So, in total, for this disaccharide, you have an alpha 1,4 linkage, or an alpha 1,4 glycosidic bond. Then you look at the next one, right, and it looks weird. First of all, you're not linking it with carbon number 4 in the second monosaccharide. You're linking it with carbon number 6. It's actually from that CH2OH that was sort of sticking up, right? So that's one thing. And also, you notice there's this little swoosh that's happening between your anomeric carbon and your oxygen, right? Where the red line actually dips down before it curves up. That's literally to indicate that, again, that OH was originally at the alpha position in that anomeric carbon. Just the positioning of trying to draw a bond to the carbon number 6 just makes it a little awkward to do that. So you have to be intentional when you're drawing that bond. You have to draw that little curve, that little dippity-do, to indicate that, yeah, you did have that alpha linkage at the original OH, at the original anomeric carbon, excuse me. So when you see glycosidic bonds drawn in saccharides, they will sometimes look kind of odd because you have to have that directional intentionality. There's another picture in your book where they took a couple of saccharides and they drew something they called beta-beta-1,1 glycosidic bonds. Well, first of all, let me tell you why we have two betas. All right, so 1,1, that means the two, when you have 1 to 1, that means that each one of the carbons in the respective saccharides is carbon number 1, aka the anomeric carbon, right? So in this case, you have two anomeric carbons that are linked to each other. Beta-beta tells you that the first one had an OH in the beta position, and it tells you the second one had an OH in the beta position. If you've got two anomeric carbons linked together, you've got to show the alignment of each anomeric around each one of the anomeric carbons, right? So we had beta and beta. And remember, in beta, the OH is pointed up. So that takes us back to this photo. Hold on. There we go. All right, and I labeled this photo, something is wrong with this picture. All right. So take a look at that and think for a second, do you see anything wrong with that picture? Maybe pause it a second so you can't spoil it, because I'm about to explain it. All right, if you look at the left side where basically it shows the two saccharides kind of drawn on top of each other like this, they drew that linkage showing on the top structure the red line doing a little curve up before it comes down. That indicates that you have a beta OH on the anomeric carbon. On the bottom, they show that linkage, that red line coming up. That also illustrates that it has a beta linkage at that anomeric carbon. So that actually looks pretty good to me. Where it goes sideways is in the second picture, where it shows the side by side. Right? Now, in order to show it side by side, they actually had to take the second monosaccharide and sort of flip it upside down. So you'll notice it looks kind of upside down relative to the other one. The part I want to highlight is on the left ring, anomeric carbon, you see the red line going down. That is incorrect, right? If the red line goes down from the anomeric carbon in this form, what did we say that is? We said that's an alpha arrangement. So you actually should have had a little fish hook on the left side that went up and then down, instead of just slanting straight down. So that is what is wrong with that one. Here, just to avoid any confusion, I'll draw what it should have looked like over here. So just give me a second. Okay, so this one's okay. The line's pointed down here, but remember, we had to flip our entire structure upside down, so this is actually correct. But on this side, you really should have done something like that to properly show that you had a beta arrangement. So that was a little bit of an oopsie there on the part of the book in that particular picture. All right, if you go on down, you can see, you can make more than one saccharide. You can have a chain that has more than two saccharides in it, right? So And you can make different types. So these are actually just two different versions of arranging glucose. There's nothing but glucose in these photos. In part A, you've got like 5 of them here that are linked together in just a direct head to tail chain, sort of. So these are all alpha 1,4 linkages. Alpha 1,4, alpha 1,4, alpha 1,4, alpha 1,4. Right? Then you kind of see the same thing in the second one, but you can see, you know, these rings have more than one OH, so they can make more than one glycosidic bond. So you see a sort of that linear chain of alpha 1,4s, but you also see some alpha 1,6s that are coming off, is what they call branch points. We'll get more into that in just a few minutes. But first, let's highlight some common disaccharides. You've probably heard of a lot of these. All right. Lactose. What is lactose? Well, that's a milk sugar, right? Commonly found in milk. Lactose is made as a disaccharide made out of galactose and glucose and a beta 1,4 linkage. Sucrose, that's probably the most well-known sugar. That's table sugar. That's the most common sugar that sweetens everything that is sweet, right? It's in ice cream, it's in cookies, it's in cake, it's in candy, it's in whatever. Sucrose is actually made out of one glucose unit and one fructose unit in an alpha 1,2 linkage. Maltose is a sugar commonly found in malt beverages such as beer, made out of 2 glucose units and an alpha 1,4 linkage. Excuse me, I'm struggling over here. Celobiose, I'll be honest, I kind of forgot what celobiose does, so you'll have to look it up. But glucose, 2 glucoses in not an alpha 1,4 linkage, a beta 1,4 linkage. But you can see it's very similar to maltose, but even just that slight difference in having a beta versus alpha linkage makes a big difference in what it can react with, what physical properties it has, how it interacts with biological systems. Here's a couple of other sweeteners. We talked about sugar alcohol before. Saccharin is actually not a sugar at all. It is that structure right there. I don't really remember why I put that in there, to be quite honest, but it's there. It's interesting. I think more interesting than that is sucralose. Sucralose, if you compare the structure of sucralose to sucrose, that's in the next slide, you'll see they're very similar. Sucralose is actually a direct sucrose derivative. You've just replaced one of the OHs with a Cl. And that makes a world of difference because we can digest sucrose, we can break it down, we can get calories from it. Sucralose, you can't. Just having that one Cl in place of an OH means that we can't enzymatically catalyze the breakdown of it, so it just passes through our systems, right? You probably know sucralose better by its trade name. It is called Splenda, right? Yep. That's the only, that's really probably the only artificial sweetener I actually like. I don't like aspartame, I don't like saccharin. Sucralose, though, it tastes pretty good, I think. But that's to each their own. All right, deep breath. Let's talk about polysaccharides. Well, polysaccharides, exactly what you expect. You're gonna have lots of monosaccharides. You're gonna have lots of glycosidic linkages. And there's a variety of different polysaccharides you have. So we're not going to talk about all of them, but we're going to talk about some of the more interesting ones. First of all, some terminology. A homopolysaccharide, this is where all the units are the same. So if you have a bunch of glucose units all linked up in a row, in a big chain, that would be a homopolysaccharide. Heteropolysaccharide is going to have more than one monosaccharide. It could be something composed of, let's say, glucose and galactose, or glucose, galactose, and fructose, etc. Right? You're going to have more variety. Regardless, though, you're gonna, and this is a common thread, we see this in proteins, we see this in nucleic acids, you're gonna end up with patterns. You're gonna have certain repeating linkages that tend to happen over and over. And that tends to give the polysaccharide its identity. All right, I want to start off with cellulose here, and again, on the iPad, it kind of messed up some of my Greek letter formatting, but you can see this on the computer or the book. Cellulose is a homopolysaccharide. It's composed entirely of glucose, specifically beta-D-glucose, that are joined up in beta-1,4-glycosidic linkages. All right? When you think of cellulose, what do you think of? What is cellulose? I'm sure you've heard the term before. Think for a minute. See if you can remember where you've heard that before. Cellulose is a structural polymer in plants. It is present in high amounts in stalks and stems. Wood. Wood is cellulose, right? Tree wood. So, very much a structural polysaccharide. And that means they're going to have a lot of strength. They're going to have a lot of durability. It's hard to mess up their physical structure without a lot of force. Why is that the case? Well, it just so happens when you have beta-D-glucose in these beta-1,4 linkages, it tends to optimize the arrangements of groups for hydrogen bonding. So you would get hydrogen bonding in any polyglucose chain, but these are optimized. The groups are a little bit closer together with these linkages, and though the strength of hydrogen bonding is maximized more than it would be, and that gives it extra oomph, right? It gives it extra strength in staying together. It also means that we can't digest it, right? Any of these sugars are going to be dependent on interacting with certain enzymes to catalyze their breakdown, right? For cellulose, you have to have there's a class of enzymes called cellulases that we do not have. So, we can't eat wood and digest it. I don't know too many people who are trying to eat, a piece of wood, but if you would, you wouldn't be able to digest it. We do eat stalks and stems, right? We eat, lots of plant matter, salads, vegetables, etc. Right? A lot of times you hear it referred to as roughage, and it can go right through you, and that's because you just simply cannot digest it, right? So roughage is essentially cellulose. There are certain things that can digest cellulose. Think for a second. Do you know of anything that can digest cellulose? Do you know of anything that can eat wood? I'm sure, hopefully, maybe, the answer termite came to mind. So, termites must have cellulases, right? Wrong, they do not. However, in the gut of termites live certain bacteria that do have cellulases. So what happens is the termites will chew up the wood, swallow it, and then the bacteria that are present in their gut will actually digest it. So, kind of a symbiotic relationship there, I guess. But we can't digest it. All right, and there you see the structure of cellulose, a portion of the polysaccharide chain. We, as animals, do have... Oh, excuse me, sorry, I'm skipping ahead a little bit. There is another type of plant-based polysaccharide that's glucose-based, in fact. That's a little bit different. It's called starch, and I'm sure you've heard of starch before, right? What are some common foods that are high in starch? Think about it for a second. You probably know some of them. Potatoes, high in starch. Corn. I'm actually not sure which other ones. The beets have a lot of starch? I'm not sure. Anywho, yeah. There are things that have lots of starch. Starch is very similar to cellulose, but it's also notably different. Just like cellulose, it's basically just a linear chain of glucose. Excuse me. However, the linkages that you find in linear starch change is going to be different. Whereas cellulose has beta 1,4 linkages, starch in its linear form has alpha 1,4 linkages. And this actually, the linear starch has another name that's commonly referred to, and that's called amylose. All right, so the amylose form of starch, alpha 1,4 linkages in a linear form. So very, very similar to cellulose, but you have an alpha linkage instead of a beta. That makes a big difference because starch is definitely not a structural polymer. It has much less structural integrity. And in fact, we have enzymes that can break down starch. And that starts in your mouth, in fact. There's something called salivary amylase that can actually start breaking these glycosidic bonds as soon as you start chewing up a starch-based compound in your mouth. All right. There's another form of starch that has branch points. So we showed you that photo earlier where I was kind of leading in, right? You get linear polyglucose chain and also branch points. Starch is a good example of that. All right. Whenever you have these starch chains that have branches in them, first of all, the branch points are going to occur typically at alpha 1,6 linkages. And this is referred to as amylopectin. We can also break down amylopectin. You don't use amylases, though. You use something called a debranching enzyme. And the name is pretty self-explanatory there. But we do have the capacity to break down both forms of starch, and we can digest it, and we can get calories from it. A little off topic, but just too fun not to include. You can do a very easy test for starches by adding iodine. And this, you can just literally get the iodine solutions from the pharmacy, the ones that, in the old days people used to use as antiseptics. They still sell it, even though it's not really the preferred antiseptic anymore. But anyway, if you add starch and iodine together, what happens is you can form this very, very rich dark blue color. It's kind of brilliant. And it's actually formed because iodine can fit into the helix of amylose. Amylose will actually curl up and form a helical structure, and so iodine just happens to fit in perfectly in here in complex with the starch, and when it complexes with it, it forms a dark blue color. So I remember when I was a kid, that was actually a fairly common crafting thing. You could actually do something like cut a potato in half and then like cut a design in it, like with a knife or something, and then put that into a solution, sort of put it into a starch iodine mixture, and you're sort of creating your own rubber stamp. So it's kind of cool. Fun little craft to do with the kids, or even on your own. Fun fact, that blue color, you can completely make it disappear by adding vitamin C. Vitamin C actually breaks up that complex, so it makes it go clear again. I've done that demo a lot of times. It's a lot of fun. Not really applicable to you guys, but, just some free information. Alright, so I told you about the enzymes. Let's talk about glycogen. So glycogen is very, very, very similar to amylopectin. Just like amylopectin, it has sort of a baseline of glucoses and alpha 1,4 linkages, and then branches where you have alpha 1,6 linkages to glucose. So that's the same as amylopectin. The difference is that the branching occurs more often in glycogen. You get a branch point about once every 10 residues in your linear chain in glycogen, as opposed to about once every 25 units in amylopectin. The other big difference is where these are found. Starch is plant-based; it's only found in plants, but glycogen is actually found in animals, right? Specifically, it's used to store extra glucose in animals. You'll see a high level of it in the liver. I don't know if any of you guys eat liver. I can't bear the thought of it because it just seems gross. But my parents love it, and I know a lot of other older people love it too. So, but anyway, I've heard that you get some liver from the butcher and you cook it up. It actually kind of has a sweet flavor to it. That's what I've been told. That would be from the stored glycogen that's present in there. It has lots of glycogen present. And so basically what happens here is we have extra glucose that's not being used by the body. It gets shuttled into glycogen storage, and then when we need it, we can go in and snip off glycogens or snip off glucoses with glycogen phosphorylases, which we mentioned in a previous chapter, and also debranching enzymes. So very similar to starch, but they're found in different places. And slight difference in branching. All right, just want to highlight a couple of others here that are pretty interesting. Also want to emphasize here, the ones that we went over, the monosaccharides we talked about before that were, the common ones, glucose, galactose, fructose, allose, etc. Those aren't the only ones. You can have, just like with amino acids, you can have all sorts of little derivatives and stuff that exist. So some of these other ones you're going to see ones that we haven't heard of yet. I want to talk first about something called chitin. You may have heard of chitin before. Chitin is basically the exoskeleton that exists in a lot of bugs, insects, other creepy crawly critters, lobsters, crabs, the shell stuff, not turtles, but other shell, like, what do you call them? I'm showing my lack of biology knowledge here. I don't know if it's not cephalopods, but anyway, sorry, I'm embarrassing myself. Let's just say examples: insects, crabs, lobsters, etc. Beetles, that's another one. This is composed largely of something called chitin. And chitin actually is composed of a particular monosaccharide called N-acetyl beta-glucosamine, joined up in beta-1,4 linkages. And so if you scroll to the next slide, you can see the structure of that particular sugar. This one's a little different. It looks sort of like some of the other ones, but this one actually has an amide linkage at carbon number 2 here. And you can see you get continuous linkages beta-1,4. If you know anything about chitin and these exoskeletons, is that these exoskeletons are designed to protect. They're very durable. They're hard to break. They offer the creature protection against the elements, its surroundings, predators, etc. Right? If you've ever eaten lobster or crab, you have to get the mallet and you have to crack it. Right? They have a lot of structural integrity. That's because this one has really optimized hydrogen bonding that can occur. It's amazing the difference that hydrogen bonding can make in determining strengths of different compounds and structures. The amide groups that are there, the NH groups, and the carbonyl oxygens that are hanging off, offer extra opportunities for hydrogen bonding that aren't there in a lot of the other sugars. So that's why that one is so much more structurally sound. There's another group called peptidoglycans. And again, we saw this with lipids. You can have a mixture, a mingling of different biomolecules together that can make a structure for an intended purpose. Peptidoglycans, again, they're used in bacterial cell walls to help provide a layer of protection from anything that might damage the cell. This is actually a mixture. You have a mixture of polysaccharides and peptides here. All right? So basically you have polysaccharide chains, all right? And these polysaccharide chains are made out of a couple of different saccharides. One's called N-acetylmuramic acid, or NAM, and N-acetylglucosamine, NAG. All right, and they're joined in beta 1,4 linkages. So if you look in part A over here, you can see a couple of these units that are actually linked together. And then they are what we call cross-linked by peptides. So we've talked about peptides before in chapter 3, right? So you remember what they are. You can have these little amino acid sequences sort of hanging off these saccharide units, and what they do is they tie basically one polypeptide chain, one polysaccharide chain to another. They act as bridges that connect these polysaccharide chains. That's where you get the term cross-link. And if you're ever to take a polymer chemistry class, you'll learn that cross-linking is a highly effective way to give a structure extra added structural integrity, strength, and durability. So again, this has been optimized in bacterial cell walls to provide good protection against whatever might damage the cell. Similarly, plant cell walls have things that can protect them. Cellulose is a large component. And also another one that you may have heard of called pectin. All right? Pectin, the structure of the monosaccharide that makes pectin is found right here. It's called D-galactouronic acid. Excuse me. These are linked up in a chain. Pectin has similar properties in a lot of ways to cellulose. It provides good structural integrity and strength. We were talking about quote-unquote roughage before. Pectin is also included in that term. Pectin and cellulose are the two primary components of typically what they call roughage. And then the last one I want to mention is certain types of what we call glycoproteins. And you can kind of figure this out from the name, right? Glycoproteins, you're going to have polypeptide chains, but you're also going, polysaccharide chains, but you're also going to have polypeptide chains. There's going to be very much protein-based. So actually, typically what these are usually is that you have a polypeptide chain and you have saccharide sequences that tend to, I guess you could say, cap the ends of these polypeptide chains in various patterns. They're commonly found, used as antibodies. And probably the most common application of this is in blood typing, right? I'm sure you guys in blood type heard about different blood types, type A, type B, type AB, type O. Basically, what is happening here is that, excuse me, you have these polypeptide chains, these protein chains. And at the ends, you can find certain sugar caps, right? So, if you look at these glycoproteins, there's one particular sugar called alpha L-fucose that's always found at what we call the non-reducing end of the chain, right? So that's always there. However, different glycoproteins will sometimes have other things in addition to that. If you have blood type A at the non-reducing end, you're also going to find the presence of N-acetylgalactosamine. I don't remember if I put the structures in here or not. Yeah, here we go. So, here is, on the top here is a type A blood type at the non-reducing end. So you can see several different, saccharides here. Here's your alpha L-fucose. Here's an N-acetylgalactosamine, here's a galactose, and then at the very end of your non-reducing end, you have beta N-acetylgalactosamine. Ooh, that's a mouthful. So it's there. It's highlighted. That's always present when you have type A. However, if you have type B, you don't have that one at the very end. You have alpha galactose instead. Right? So it's the difference of those two polysaccharides that makes the difference between type A and type B. If you have type AB, you got both of them. If you have type O, you don't have either one of them. Right? So, that's the difference whenever you're looking at different blood types. Is literally you're just looking at the presence or absence of two particular monosaccharides capping the end of a polypeptide chain. All right, that ends chapter 16, which is good because I'm on the verge of losing my voice. I will post this one and the other one shortly, and I'll let you guys know. Well, why am I telling you this? If you're watching this, you've already seen them, so never mind. But if you have any questions, just let me know. All right, till next time, I'll talk to you later. Bye-bye.